So this is going to be the lab video, biolab video for, it's called the macromolecule or organic chemistry. So, there was 3 videos I made for this lab, and I'll be just making 1 video for this lab, this video here. And then next week, or for the next video, I'll make an enzyme lab video for that lab. So this is the lab PowerPoint. All right, I told you it's a good idea to study the lab PowerPoints for the quizzes and for the practicals. Okay, so the organic chemistry lab really is, the macromolecules is just another name for them. They're all organic molecules. In this lab, what we would be doing if we had a lab in face-to-face would be to complete tests to detect lipids, proteins, and carbohydrates, right? So you're going to need to know the names of those tests, what those tests test for, what a positive result looks like when you take the practical. I might ask you specific questions about proteins and carbohydrates on the quiz, and I'll go over that material. So we talked about this in class as well. Carbohydrates is one of the 4 macromolecules, and they consist of sugars like glucose and fructose. Sucrose is table sugar. A monosaccharide means there's just one ring, one sugar. If it has 2 sugars, like sucrose, you've attached glucose and fructose together to generate sucrose, then it's called a disaccharide. If you have starch, starch is many glucoses, up to 1,000, attached together to form a polysaccharide instead of, right? So you have mono, disaccharides, polysaccharides means many sugars. So starch, cellulose, and glycogen are examples of polysaccharides that all contain glucose. They're only showing, this is supposed to be a glucose, each one of these. They're oriented differently. You don't need to know the differences, but they're made of the same thing, but the glucoses are linked together differently to make them different molecules. So plants make starch, animals make glycogen. Cellulose is made by plants as a structural component in their cell wall. So when you put these sugars together, whether it's 2 or many of them, you use a glycosidic bond that's a type of covalent bond. They're all linked together. So each one of these hexagons is one glucose. There's as many as 1,000 of them in each molecule. In the liver, you might have just trillions and trillions of glycogen molecules. It's about 10 grams altogether. But fruits and vegetables contain starch. So lipids is another example of it's macromolecule number 2, I guess you could say. And these are examples, so you should recognize these as types of lipid that are hydrophobic, right? They don't like water. Okay, amino acids make up proteins. So proteins, macromolecule, you polymerize amino acids to make them. When you put amino acids, when you link them together or polymerize them into one long chain, it's called a peptide chain, you generate peptide bonds between the amino acids. So you don't need to recognize this information here. It's just showing you how this was an amino acid and this is amino acid. The plus means they're together, they're separate, and you connect them. They're not connected by a peptide bond. So these are the tests we would be performing in the lab, right, if we were doing the lab face-to-face. The Benedict's test is a test for simple sugars. Simple sugars, this means they're small. They're reducible is another way of putting it. The Biuret test detects proteins. You get a violet color. Benedicts, you get an orange color. Starch, when you add iodine to starch, it stains or dyes the starch really dark brown. Sometimes it even looks dark blue, dark or almost black. It just depends on the starch. If we want to detect lipids, there's two methods. One's called paper spot. The other one is we use a dye called Sudan-4. So you should recognize the names of these and what they detect. So this is a test tube containing, there's no sugar in this test tube. There's sugar or reducing sugars in this one. You add the Benedict's reagent to do the Benedict's test. So we would have added, 10 or 15 drops of Benedict's solution. We would have put it in boiling water. The negative, you get no orange color, and you get like a nice tan color if there's reducing sugar or simple sugar present. So on the practical, you might see this image, and then I'd expect you to tell me which one's positive, what's the name of the test. So those are the kinds of questions. It tests for what? Right, simple sugars, reducing sugars. This one's a little bit harder to see. The negative tends to be this blue color. So for Biuret, you add Biuret reagent, and you get a violet color if protein is present. I'm not quite sure what this one is here. Just ignore that. This would be a negative test. This would be a positive test for Biuret. That means protein is present, no protein is found in that test tube. Now the Sudan-4 is a red color. If you have oil in a test tube, this is not a very technical one, but 2 layers means you have oil on top probably and water on the bottom. It turns a red color, but just having 2 layers really tells you it's oil. The red color kind of helps confirm it. But you should recognize Sudan-4 as a test for lipids. These are actually vegetable oil that we used in the lab. This is supposed to be a piece of bread here. It's hard to see. And what you'd have done in the lab is add iodine to the bread. If it turns this dark shade of black or blue color, then it's positive for starch. You can add this to a potato. Potato contains starch, and you get a similar result too. So the other name for this is Lugol's iodine or iodine. Right, but I just put iodine here. The other word that you could see, like maybe on the handout or something, is Lugol's iodine. It's Lugo, G-O. So you might see that word used, but there's always iodine will be associated with it. And then the other part of this lab was to look at what's called the DNA puzzle. Right, so the DNA is another macromolecule. There's not a test for this specific molecule, right? We wouldn't have done a test for it in lab. We would have had you put together these puzzle pieces. So there's a phosphate, which is a yellow puzzle piece. There's a deoxyribose puzzle piece that looks pink or red. And then the different nitrogen bases have different colors depending on which one it is. But if I say, what's the yellow piece on a test? Phosphate, correct answer. What's the red? Deoxyribose. And what's the green piece? If you just put nitrogen base or N base, that would be enough. Base wouldn't be enough. N base means N stands for nitrogen, so nitrogen base would be correct as well. These are the different, but unfortunately this isn't colored. This is white, but they're all different colors. The T and A and G and C, right, tell you which base it is. Now this would be a DNA puzzle of basically one strand here, one strand here. They're held together in the middle by hydrogen bonds, right? So a single strand of DNA would be, if you pulled these two apart. So we, in lab, you would have put this puzzle together. You would have only used 4 bases instead of, this has 8 altogether. But this is a nucleotide, this is a nucleotide, this is a nucleotide. This phosphate's attached to this nucleotide. You see how the phosphate's attached here, not here. On this end, the phosphate's here, not there. So this phosphate goes with this nucleotide. So putting that puzzle together helps you see that, and that there's ends to the DNA. This is the 5' end, this is the 3' end, and we'll talk about that, what that actually means when we do the DNA chapter. It's like chapter 16, it's pretty far away. Now the RNA puzzle, right, means that this is ribose instead of deoxyribose. You'd only see U in RNA, you only see T in DNA. So with this, the OH is what you get. You see this OH here? That's an O and this is an H. You see it kind of, it's all the letters are actually from this side. OH, you don't have that OH, you just have a hydrogen here. And that's the difference between the two. The small point, I'm not going to expect you to know those distinctions. If I had a puzzle piece on the practical, it would just be, say, 1 phosphate, 1 sugar, 1 nucleotide, and if you got as long as you said ribose or deoxyribose for this puzzle piece, it would be, I would mark it right, or I wouldn't expect you to be able to tell the difference between the two. So this is the enzyme lab, we'll do that next time. This is the big difference between DNA and RNA. DNA has deoxyribose for a pentose sugar, RNA has a ribose. These are the bases. They all have A's, G's, C's. DNA has T, RNA has a U. Base pairing. We'll talk about this in the DNA chapter. Don't worry too much about it for this purpose. And DNA consists of 2 strands. Most of the time, RNA is single strand. Okay, so I think you'll be, there'll be a quiz that'll have questions from the lab number 1 and this lab. Okay, and be sure you complete it. Quiz 1 covers labs 1 and 2. The enzyme lab will be on quiz number 2, as well as the cell lab and diffusion lab. Okay, so you can use this schedule to help see when and what's going to be on the quizzes. Okay, so anyway, study hard.